Unless we respect and recognise the importance of a fungi, if there's no fungi, there's no plants. If there's no plants, there's no us.
Food. No fungi, no food.
Well, and no us.
And no us, yes.
Yeah.
Hello and welcome to the On Farm podcast. It's Anna with you this week. It's definitely a not to miss this episode because hugely interesting and educational. But I shall let our guest introduce himself.
My name's Neville Kilkenny. I'm a consultant mycologist. I do field work and laboratory work to support scientific projects and environmental projects.
Right, so where are we off to now? Tell me why we've got in the pickup and where we're heading.
So we're just going to go down to the end of the farm road where the wood from Cross Hill meets the road there. It's actually quite an old bit of woodland. We're just going to have a look and see if we can find anything that might indicate the age of the wood and things like that.
Okay, cool. On the way there though, can you tell me a little bit more about what you do? I've been on your website, which I've got to say is the coolest web domain I think I've ever encountered. Can you tell me what it is, and then tell me that might give people a clue. But then tell me about your day job.
So, fungi.co.uk is basically it's a point where I can just let people know about the public events that I'm doing. That's a combination of sort of foraging style events or just general sort of biodiversity, telling people about fungi and raising the profile of fungi. But the main bulk of my work is actually surveying for sort of land management advice. For instance, we're doing a big project with RSPB at the moment, looking at different types of woodland management to see what types of interventions protect or encourage the greatest biodiversity. Mm-hmm. And we do a lot of environmental impact assessment as well. Particularly at the moment, there's a big drive to plant trees, obviously, because of climate change. And there's a loss of habitat that's unrecognised as important. Although it's not important for vascular plants, for fungi, it's very important and underappreciated. And there's a lot of unimproved grassland, for instance. Although we're very rich in these grasslands, in the European context, these grasslands are very rare. So we're the stronghold for these unimproved grasslands across
Scotland is that stronghold, and so
well, Scotland and England actually.
Okay, Are people using that as a sort of a benchmark and something to learn from?
It's more a case of what we don't want to do is with this huge drive for people to plant trees, is to plant them in appropriate places. And one of the obvious places is these grasslands, which, from a vascular plant point of view, look very uninteresting. But actually, because of a lack of intervention or any soil improvement or anything like that for an awful long time, these are really rare habitats.
Yes, yeah. And so the uninitiated would say, what's so important about fungi? Why should we care? Why should we be interested?
Look out the window. None of this would be. I mean, basically, we've got two types of fungi. They derive their nutrition either by recycling dead organic matter, and without those fungi recycling, the sort of 460 billion tonnes of dead organic matter that gets deposited on the land surface every year, we'd be absolutely drowning in the stuff, and none of this around us would be able to grow. So the fungi form the base of our food chain by recycling all this organic matter. And 90% of all plants require a fungus on the roots to be able to grow and take up enough nutrients from the soil to be able to thrive.
I did not know that. Wow. So I suppose, like much of what happens in farming and in rural areas and woodlands, a huge amount of this is not visible. We're the naivety of people like me thinking that you can see it. As you say, the majority of the activity is totally invisible to us and going on completely out with our awareness.
The statistic that I love is that they say that in a teaspoon of healthy soil, there's 6 kilometres of fungal hyphae. If all joined together, so joined together, would reach from here to Haddington in a teaspoon of soil.
No,
yes,
that's unbelievable, isn't it?
Which so when we look at the importance of fungi, if we consider the potential biomass of fungi if we restored all our soils back to health. And you think about 50% of the planet's soils are depauperate and not healthy.
Yes.
If you think about the fungal biomass, if we restored all those soils back to health, the amount of carbon capture within that is vast.
That's phenomenal. So what are we doing that we shouldn't be doing? And what should we be doing more of in terms of taking care of this and ensuring that that the fungi has the ability to continue to develop and thrive, and support, that continue to be a necessary support structure for everything that we see.
So it's really recognising habitats and understanding why things are where they are. And one of the things we find, for instance, in our woodlands is that large diameter deadwood is one of the first things to leave the forest, because everyone recognises it as a resource, whether it's fuel or for construction or whatever.
Yes.
And so our woods generally are massively lacking in deadwood, and especially large diameter deadwood. One of the things that fungi really suffer from is nitrogen in sort of acid rain and pollution from cars, and I'm afraid the runoff from agriculture. These are huge threats to fungi.
Yeah. And I'd love to talk, we'll get out of the car in a minute, I'd love to talk about the connection with agriculture, because I think that's critical. Right, so we're out of the pickup, into the wind, and heading into this wood. So I'm going to let you take over, I think, and just tell me where we're going, what we're looking for, what we're looking at.
So the first thing we're looking at is the assemblage of the vascular plants we've got here. We're looking what sort of trees we've got. We've got ash trees, we've got sycamores. These are the type of trees that associate with what we call arbuscular mycorrhizal fungi. So we won't find toadstools associated with them unless they're litter rotters. And this is one of those litter rotters. This is what
litter rotter.
Yeah.
Right.
So this is what's called a cloud agaric.
Yeah.
Or Clitosomy nebularis. Typically, Clitosomy, the funnel caps, this group, they have gills that come down the stem, they have a white spore print. And spore colour is very, very important. The starting point of separating all the different gilled mushrooms. Mm-hmm. And the gills are just one example of a reproductive surface. The reproductive surface is where the fungus is going to make its, where the fruiting structure, where the spores are formed.
Yes.
And typically we have a reproductive structure, a cap to protect it, and a stem to lift it above the surrounding vegetation so it can drop spores into the moving air.
Gosh, yes. All the function that you wouldn't think about when you're just in a wood looking at this. Yeah.
So the spore colour, and in this case it's white, and the spores range from white to cream to pink to milky coffee brown, to a rusty brown, to a chocolate brown, to a purpley black, to dark black, and we get some fungi with green olivaceous spores, and in the tropics we even get ones with blue spores. Mm-hmm. So the spore colour is the starting point of identifying our fungus. And if you look at the mushroom guide, you tend to see the ones at the beginning are ones with white spores, and they slowly work back to the ones with the black spores at the back.
Okay,
and then they go on to other types of fruiting bodies.
Yes. So before we talk about that in more detail, because that fascinates me, we've picked this gentleman or lady up from the ground. What have we interrupted? What was it actually doing down there that's beneficial for what we see around us?
This is literally just the fruiting structure, so this is like picking the apple off the apple tree. Mm-hmm. It's not actually hurting the organism whatsoever.
No.
In fact, we did a little experiment down at Doit Botanic Garden where we had a patch of chanterelles, and half the patch we picked, and every year it came back with a flush of nice toadstools every year, and the other side we didn't pick, and you could see actually it slowly waned, and we think maybe the fungus died.
Oh goodness!
So,
right. So
it could be a little bit about pruning a plant.
Yes,
that you make the growth more sort of vigorous.
Like my dahlias that I grew this summer. The more you pick, the more they flower, and if you just left them, you wouldn't get nearly as many buds.
With the proviso that obviously, if there's no fruiting bodies, if there's no flowers, the organism can't reproduce.
Of course, yes.
So you can't take everything. The fungus itself is in this upper soil horizon, and it's recycling all of this leaf litter. This particular one is always a late fruiter. You always see it towards the end of the year.
Okay.
And this actually, everything this year is very, very late. And this is one of the things we've noticed. A very good indicator of climate change is that fungal fruiting, the period from the start of the season to the end of the season, has almost doubled in the last 70 years.
Oh gosh, right. So we've come across, I'm not going to move too far because I don't want to damage anything, but we've come across this couple there. Now these look similar but different to what we've just seen over there.
So they're in the same group, the same genus. It's got a little point in the centre of the cap there.
Oh yes.
I'll pick one of them for you. And again we have the gills that come down the stem.
Yes.
So again it's a Clitosomy, it's a funnel cap. And this one is what's called the trooping funnel. and this one that is considered good and edible. And
Okay,
a lot of
yeah.
people eat this one.
And actually, do you know that they're beautiful, aren't they?
Absolutely stunning.
I'm going to have to confess here that I don't like mushrooms of any sort. In fact, my husband made mushroom soup yesterday, and I was like, "Oh, great, thanks."
Do you like chocolate?
I like chocolate. Yes.
What if I told you that chocolate wouldn't be possible without a fungus?
It makes me more appreciative of what I'm looking at.
So the cocoa bean's very, very bitter when it falls from the tree.
Okay,
But it's
yes.
only as it starts to rot, when it starts to be decayed by a fungus, it develops a sweetness.
Wow,
And
goodness.
it's the same with coffee beans as well.
Yeah.
And you'll find actually in your supermarket shop, nearly everything you buy from the supermarket will involve a fungus at some point, and it's either as an ingredient or in the way it's produced or in the way it's packaged.
It's fascinating, and whilst I can't appreciate that, the taste of that, maybe I am for the first time also appreciating its beauty and its intricacy.
One of
it's
the key characters
fascinating.
of fungi that nobody ever really seems to be aware of is that fungi tend to smell. This has got quite a flowery sort of perfume scent to it.
Yeah.
But you get ones that smell of aniseed, some smell of marzipan,
Really?
some smell of leather.
There's a gentle softness to that actually in the smell.
So smell is a very important character, and this is one of the things that always terrifies me about these apps, is that so much of fungal identification is all about texture and smell, and how robust a fungus is, whether the stem's hollow or whether it's firm and things like that. And of course, these apps are just looking at pictures effectively. And I mean, the AI is very good. And actually, I mean, if you look at some of the larger sort of AI or more developed AI products that have access to thousands and billions of pictures, then sometimes these apps can be spot on. But in general, apps are not great. I mean, fungi have no respect. With the fruiting structure, they have no respect for size, colour or shape.
No. And how can an app replace, as you say, the sense of smell? You know, for you, that's a fundamental identifier, and you can't replace that. This is a very broad question, and we don't have all day. But this is just really making me think. You know, what? Going back to agriculture and food production, you know, as you said, that huge proportion, the vast majority of the food we buy in the supermarket wouldn't exist without fungi, including my favourite chocolate. But what do you think agriculture can learn from a deeper study of fungi?
I think we need to invest money in more research of fungi. Mycology in Europe, I was going to say in Britain, but right across Europe is in crisis. No one's funding any taxonomy. There are fewer and fewer mycologists in post, and if they are in post, they're not just working on fungi, or they're too busy teaching to actually be able to, work on fungi. Also, we've now discovered fungi that can degrade plastics, fungi that can make biofuels, microremediate oil spills, things like that. So there's masses of applications of where fungi can help. And again, with our carbon footprint, I mean, fungi in general are one of these organisms which have huge potential both in construction. You can use fungus for packaging, insulation, you can laminate boards instead of using glues. You can make bricks out of fungi, potentially you could make car parts out of fungi.
Oh my goodness.
And then, of course, it's all biodegradable.
Yes.
And what's more important is it's a circular economy. Let's say you've got your agricultural waste, you've got all your husks from your cereals, you then can grow a fungus in that, you could potentially grow a crop of mushrooms from that, and then that fungus can then, that organic matter, the fungus can be allowed to grow through it completely and then into a certain shape that you can use as a brick or a component or as a block of insulation or whatever. You cook out the fungus, and if you do it under pressure, it can be an incredibly strong
This
structure.
is fun. This is just phenomenal. We've created about 170 episodes of this podcast, and I don't think my eyes have ever been opened to this extent by any other episode. This is just mind-blowing.
But also, I mean, going back to what we were saying about soil restoration and the potential of carbon sequestration through fungal biomass in
very
soils.
important.
What we're doing to our soils at the moment, the constant disturbance of soils, is really detrimental to fungi. And the lack of organic matter within the soils as well is hugely detrimental. I guess the more we can move to processes where there's a minimum tillage, intercrops planted between crops and sown back into, putting organic matter into the soils, that sort of thing is always going to help fungi. One of the things that really stands out is also with this huge drive to plant trees, a lot of farmers are turning to that as a source of income. Because there's a lot of companies wanting to offset their carbon, and the easiest way is to buy land, easy for them. Whether it's agricultural land or unimproved grassland or whatever. Buy that land, plant trees, and banks are prepared to lend them money for that. It's a good investment and like that. But those trees, if you plant straight into an agricultural field, especially if they're trees that associate with fungi that make toadstools, so the ectomycorrhizal fungi, those fungi aren't there. So those trees will just die.
Will not survive. What a waste of time and money.
So I mean, and there are companies now that are thinking ahead and working with large forestry organisations and introducing inoculated fungi, whether on rootstock or in other applications, so they can introduce these fungi to those sites. But again, it's something people don't think about, is that these trees just planted out into an arable field are not going to live.
They're not, because they don't have what they need. Yeah. I'm wondering if some of this stuff is taught. If you go to agricultural college, are they learning this kind of stuff? Because it's fundamental, surely, to farming, but also to some of the diversified projects that people are looking to get into, like trees and forestry. People need to know this.
I mean, there are a lot of farmers I speak to that are aware of this, but I think it's only people that have done the research themselves. I'm not aware of it being taught. No,
the people who are interested and keen to learn. Yeah, goodness. Yeah, going back to the public facing stuff that you do. If I was on one of your days, what else would you be showing me? What else would I be learning if I was with you for the day?
I'd be telling stories a bit in the same way that I've told you about the importance of the fungi, the way they affect our lives. I'll be introducing some fungi which are considered really good to eat. I'll be telling about what they're doing with in the environment, why they are where we find them. And I'll say this fungus grows particularly with this tree because it's mycorrhizal with this tree, and you'll only find it with this, whereas that fungus grows with several different trees, and so on. And also, I'll be saying to people, we estimate about 2.5 million species of fungi out there. A mushroom guide that has an awful lot of species. There's Sir Roger Phillips's guide, which I think has about 1,250 species. It's just the tip of the iceberg.
Yes.
And if you go out and try and identify all the mushrooms you're going to encounter, you're just going to get disappointed because it's a huge diversity.
Yes.
I mean, again, I'm not an expert, but I would say roughly there's about 70 land mammals in Britain, about 700 species of bird, about 200 species of bryophytes and things like that.
A bit more manageable to identify.
When you encounter, there's estimated 12,000 species of fungi in Britain. Potentially the diversity could be double that.
Gosh,
We're describing new species every year. And as I say, across the world, we estimate it's about 5 million. So trying to identify everything is a bit of a no-brainer for people out at forest. So it's much better to try and get to know one or two mushrooms and get to know them really well. And sort of go away, collect those mushrooms yourself, reinforce your confidence. You can maybe bring mushrooms back to another walk and say, I found this, is that what I think it is? And you get confirmed. And when you're absolutely sure of that, you can move on to another one. And slowly you can build up a little repertoire of about 10 species. And if you know the best 10 species and know what they could be mistaken for, and you know the sort of 10 species that are similar that could potentially kill you, then to be honest, you're pretty safe foraging. The point is really just to start, and it could just be with a puffball, which is fairly safe and easy to identify. And there you are. You've already found your dinner. You've got that feel good thing. You've had forage. You've found you haven't killed yourself, and you've begun to notice the habitat where you find them, and you notice the differences, and slowly you're engaging more and more in your environment. That's the thing, is that we become more and more detached. And these are really important. When you look at the plants and the communities and things like that, they speak in volumes. I mean, you can just see, from, for instance, the nettles here. That's telling us there's a little bit of extra nitrogen coming off the fields and things like that.
Yeah.
As we go into the woodland, what we'll start finding is things like dogged mercury. You might find bluebells. You'll find herb Roberts. You'll find lesser celandine, and these are all indicators that this is ancient woodland. And then as we wander through, we'll find some old stumps, and on those there'll be bracket fungi that when we look at the brackets, we turn them over, and it looks a bit like a maze. All the sort of the pores are stretched in a sort of higgledy-piggledy way, and it's a sort of interim stage between. I always think of it as a fungus. Every fungus aspires to be a guild fungus, and they start off. A lot of the bracket fungi are poroid.
Yeah. They're trying to get to that goal.
And so the pores slowly stretch, and in that sort of evolutionary interstage, they have this maze gill structure. And you get one on the stumps in here. It's the oak maze gill.
Okay,
and so first of all, it tells us that's an oak stump. And secondly, that oak, that oak maze gill is one of the indicator species we have for ancient oak woodland. So it tells us that this is really, really old woodland. And I think the more you tap into that, the more you understand what's going on. You appreciate what's really important habitat, what should be protected, what shouldn't be interfered with.
Yes, and you know, we hear, don't we, about you can't do this because there's bats, and you can't do that because there's badgers or whatever. But actually, that's because they're seen and they're visible, and people kind of can connect with them. But people have not had the opportunity to connect in the way with the fungi, and they don't have that understanding. And so, that's what you're there for, I suppose, from in an environmental impact assessment type way.
Yeah, and I mean, we can count on one hand the number of species that have legal protection in Britain. We have thresholds of certain suites of fungi. We have ones for grasslands, we have ones for oak woods, we have one for dune fungi, and we have one for Caledonian pine forest, for instance. And we have thresholds that recognise sites as important with potential notification for sites of special scientific interest. But in general, fungi have very little protection.
Yeah.
It really worries me when you see sort of things like sort of people demonstrating because of bats being disturbed. Because you look at HS2 and on the amount of ancient woodlands that was intervened with there, and I think, well, yes, we probably did do bat surveys and mammal surveys and things like that. How many fungal surveys happened? Very few, I would suggest, if any. There aren't many of us doing it. And certainly I'm not aware of anybody being asked to work on that project, for instance.
No, goodness, that just shows, doesn't it?
And then also understanding how to, if there is no choice, if you have to go through that piece of land, what's the mitigation? How do you deal with that?
Yes,
and we've seen a little bit of translocating soils, that sort of stuff, but one of the things we've looked at is underplanting veteran trees with bait seedlings, and then lifting the bait seedlings with the root ball intact and moving that away from the development. So you keep the mycorrhizal, you take the mycorrhizal fungi away. Yes. So if you are doing compensation planting, you've got the associated fungi going with it. Similarly, the trees, if you've got large diameter trees and stuff like that, they're taken to the new site as well and laid down so that the wood decay fungi are there as well. So you're trying to keep as much of the environment, of the habitat intact.
But of course you can't do that unless you've done the survey in the first place, and you've got, presumably you need time to be able to enact that. So But
at the end of the day, that's never going to replace what was the ancient woodland or the ancient piece of grassland.
No. What type of people come along for a day with you to learn all this?
A very, very broad spectrum. We play a lot. I've got a lot of fantastic friends that are foragers, and they're really creative, and they share lots of tips of what you can do with your mushrooms. There's like a jelly ear mushroom you can dip in some exotic liqueur. You could maybe you've got buck and birch from Mary that make a really nice elder elixir, and you can marinate your mushroom in that and dip it in chocolate. Dip it in chocolate and have a little sort of.
There's probably a mushroom out there for me somewhere.
So, and they make lovely sort of potions, liqueurs, and things like that. And very, very, very versatile in the way that you use plants and fungi. And we love to play with that. And you always get lots of wow from people that are interested in the foraging side of it. And so we go from that, where we just show the huge diversity of flavours. We've imported all these spices from across the world, and actually we've got all these flavours here in Scotland. I wonder if we can find Let's just take a tiny little wander.
Yep.
I'm looking for some old hogweed. The seeds basically, they've got a flavour somewhere between ginger and cardamom. And the flavour's really intense. Several of my foraging friends make a really nice hogweed seed parkin, which is absolutely delicious.
In fact, I saw some of it in our garden this morning and I thought to myself, I must remember that that's there because I was going to use it as a part of my Christmas decorations, but now I could use it.
Well, just pick up the seeds. I mean, it's from the carrot family, and there's you know, sort of hemlock water, dropwort and things like that in that family as well. So you do have to be careful, obviously, and know what you're doing. Syrups you can take from different trees, like the round tree, the buds and stuff like that have a beautiful almond taste. I actually got a first prize in the Gifford Flower Show for my round shoot amaretto.
Oh, well done.
But also, I mean, we then take up other groups, like for instance, I work with the Ranger Service here in East Lothian and take them out and try and explain to them how they can talk to people about fungi, but without having to identify them. You know, again, telling stories and telling about the relationship they have with plants or the importance of them and so on and so on. Yeah. Or how they. So
that's kind of a sort of an educational remit, I suppose, as to what you do to ensure that other people can kind of spread the word.
And then it goes right from that to people that actually want to learn to identify fungi, and we do workshops at the Botanic Garden where we bring people, you know, indoors and start keying things out, introduce them to using microscopes. Because a lot, you know, if you go for a walk with somebody and they're saying to you, oh, this is this fungus, that's that, and they're naming everything they encounter, they're probably telling you fibs. A lot of these things actually need microscopic examination to be able to get to species level. Foraging has a big role to play in connecting people with the environment and people's health and well-being and things like that.
And it's a hook, I suppose. The foraging is a hook for you to get people out and for them to learn more about the conservation side as well. And if you are going to forage, do it on a small scale and don't pick everything that's there. Just pick a small proportion and I guess also try and educate yourself before you go. I mean,
there's nothing worse than seeing people posting pictures on the internet or sort of, oh, what's this? Can I eat it? And they've got this huge basket full, and you're like,
work that out before you. Yes, that's so true. That's so true. You know, you've said we've got to be very careful. You don't want to scare people, but identification is kind of key. Does it happen very often that somebody eats the wrong mushroom and ends up in a pickle?
and you know, we get calls from the Kids Hospital, or occasionally, you know, we support National Poisons Information Service about cases. I would say probably we get between half a dozen cases each year. Very rarely is it.
Is that UK wide or in Scotland?
No, we cover Scotland and Northern Ireland.
Oh, okay.
England is covered by Kew.
Yeah, so it's not vast, but at the same time, it's enough that people just need to be a bit careful and mindful.
And the thing is also that if you're eating a fungus for the first time, the degree of poisoning is directly related to how much of it you've eaten. So if you eat only a small bit and see if you have an adverse reaction, and also keep a sample in the fridge or something, because it's a lot easier to identify a mushroom rather than from your stomach contents.
Yeah, so. I bet. And as you said, different people have different reactions to certain things. So again, it's a kind of case by case thing, I suppose. And have you ever suffered any effects yourself? Or
I'm quite cautious about what I eat. I'm not hugely adventurous about the different species. I like my chanterelles and ceps and things like that, and I tend to stick to those.
Keep it safe. So now we're upstairs in your office and you've got the microscope here. So you're going to kind of just talk me through a little bit about how to identify. Yeah. So you've got, as I said, we can see the vast library. Oh, goodness, yes. And I'll show you a little bit about the process. This is a lovely series of books that have recently come out, and it's a new way of looking at things. In the traditional way of looking at keys, we've always sort of basically asked questions, what we call a dichotomous key. Okay, yes. And then we moved on to what they call a synoptic key, where you sort of were offered a suite of characters that you could cherry pick, and that would give you a code. Let's say you'd say if it's got a yellow cap, then that's A, and if it's got a white stem, that's B, and if it's got dots on the stem, then that's a G, and you get these sort of A, C, G.
whatever.
Yeah, and then you
that
can
gives
kind
you a
of
code.
work your way down, I suppose. Yes.
So, but this is a great series of books. So it's basically
Beautiful, isn't it? As well.
Yeah, you start. I mean, obviously this requires a microscope, but you look at basically the type of reproductive structure, whether it's producing spores on what we call the basidia, or whether it's producing spores in little tubes called asci, and then if it's growing on wood and roughly what the form type is, and so you can. You know, and you break out all the different form types and stuff like that.
So it's not just what it looks like; it's where it's growing as well as part of the
very
process.
much so.
Yes, yeah.
And so, but this will give you a sort of idea of the sort of characters that we're looking for. And you know, here you've got sort of, do the gills come down the stem? Has it got pores underneath? Is it a bracket?
We're standing here in front of your PC. So what are we?
So
A file called Random Fungi.
Random
Cool.
fungi, yes. So let me find a really good example of how we go about identifying a fungus. We've got a picture of a fungus here that is found in a bit of ancient oak woodland, and it's quite a rare fungus. It's called Cortinaris violaceus. The fungus here, you can see it's staining red
Yes.
in the flesh there. Here, this is the fungus in question, Cortinaris violaceus. This is the national checklist, and it says conservation status endangered, red data list, near threatened. So on and so on, and it gives you the habitat, peaty soils, deciduous woodland.
Yeah, so you've got all that information there that you can use to,
It says spores 11.5 to 13.5 by 7.5 to 8 microns, almond-shaped, moderately spiny, which you saw in the photograph.
Yes.
Presumably in deciduous with aspen or birch or beech, and it gives you the cap size and it describes it.
So you've got a lot of detail there.
And then it gives you the name of the species.
Yes.
So you work through the key and eventually you come up to a description like that.
And you pretty much get a result every time, do you? Or do
Not
you
a-
sometimes
no.
think, still uncertain about this?
Yeah, and then that's where new technology comes in. We have what's called the Bento Lab, which is a miniature little thing which you can basically you use chemistry. It's quite simple chemistry to extract DNA. You break down the cells.
Yes.
You extract the DNA. With the Bento Lab, you have the potential to amplify that DNA. You basically break down the carbon ties and into short pieces, and they then grow and expand, and you break them down again, and they grow further, and so on, and eventually you get enough base pairs. So you can send that sample with the amplified DNA away to Aberystwyth University. They'll give you a sequence. And once you get that sequence, you can blast that into a database, and you'll see whether you have a match for your species.
So somebody somewhere can always find the answer, even if you...
Yeah, and you may find it's new to science. I mean, you may find that
of course,
there is no match.
There's no unidentified as yet. Yeah, wow. This has been absolutely fascinating. Thank you so much.
You're welcome.
Genuinely one of the most interesting podcasts I've recorded, hugely educational as well, so I thoroughly enjoyed that. Thank you very much, Neville. Thank you also, of course, to our sponsor this week, so that's Bell Ingram. Thank you again for your continued support of the podcast, and thank you for listening. We shall be back again next week.